Hello students and welcome to lecture 2 of the ongoing unit Bioinorganic Chemistry. In this lecture, we will see into the mechanism of sodium-potassium pump. What is the sodium-potassium pump? It is an ongoing process of maintaining permeability of cell membrane to certain ions so that the resting membrane potential is ensured. This enables an action potential or nerve impulse to be possible at any time. So the cell membrane needs to be more negative, right? So the cell membrane needs to be more negative, that is polarized, for this to be accomplished. Also, the sodium potassium pump is an enzyme process which breaks down ATP. That means an active transport is involved. It breaks down ATP by using internal sodium ions. So energy in the form of ATP and ADP. It drives the pump to ensure the exchange of sodium and potassium ions. So 2 potassium ions are taken in and 3 sodium ions are released. This allows an intracellular charge to reach 70 to 80 millivolt, minus 70 to 80 millivolt, or the resting membrane potential for the environment, so that action potentials are occurring with no hassle as such. Now the sodium potassium pump also functions independently of nerve impulses or action potential. So it has the capacity to perform independently also. Such action potential or nerve impulses get triggered due to a stimulus. Now the sodium potassium pump allows permeability to external potassium in exchange for intracellular sodium ion. This maximizes the cell potential so that a sufficient stimulus can trigger an action potential or a nerve impulse. Now let us look at the structure and function of the sodium potassium pump for active transport. It involves basically 5 processes. It is a repeating step of a few steps. So let us look into these steps one by one. In the first step, the interior of the pump binds to ATP and it takes up 3 sodium ions in the binding site. In the next step, the ATP transfers a phosphate group from itself to the pump. So there is the process of phosphorylation that occurs. Phosphorylation occurs, which causes the pump to change its shape. So the pump changes its shape and the interior is then again closed. In the third step, the interior of the pump opens to the outside of this pump. So the interior opens to the outside and 3 sodium ions that were taken up in step 1 here are released. They are released in this third step. Then again 2 potassium ions from outside. From outside 2 potassium ions can enter and attach to the binding site. As soon as the sodium ions, 3 sodium ions are released, 2 potassium ions are taken up by the pump and they enter into the binding site. Then the binding of potassium causes the release of phosphate group, that is dephosphorylation. So in this step, dephosphorylation occurs. That means the binding of potassium causes the phosphate group to be released, and this again causes the pump to change its shape, also known as conformational change of the pump. So there is again a kind of conformational change that occurs in the pump so that it changes its shape to the original form and can be open to the inside. Now it is again open on the inside. The interior of the pump opens to the inside and the 2 potassium ions are released. Again sodium ions can enter and bind to the pump, and the process is repeated and it goes to step 1. So as we see, in the first step 3 sodium ions bind with the help of phosphorylation from ATP, right? Then the conformation changes, conformation of the pump changes, and the sodium ions are released to the extracellular space outside of the cell. The sodium ions are released. As soon as the sodium ions are released, 2 potassium ions bind to the pump and this binding causes dephosphorylation. So the phosphate group is released. As soon as there is dephosphorylation, the conformation returns to the original form. Conformation returns to the original form, and the potassium ions are again released in the inside of the cell. So once this happens, in the fifth step again, the sodium ions can enter into the pump and repeat the whole process once again. So this is how the sodium potassium pump works. Now let us look at it in a very crisp manner. So it involves the active transport of sodium out of the cell and potassium into the cell. This process occurs against the chemical gradient, and that is why we need to use ATP, because this process is happening against their electrochemical gradient. Now for every 3 ATP, 3 sodium ions are sent out and 2 potassium ions come in. So in this entire process from step 1 to step 5 that we just discussed in the previous slide, as you see here, in all these steps we see that there is involvement of 3 ATP to transfer 3 sodium out and 2 potassium in. Correct? So as you see here, in the first step it binds to 3 sodium ion from cytosol. In the previous case we were looking at an axon, which is a part of a neuron or nerve cell, right? So in this case we are seeing cytosol, it is the inside of the cell. So it binds to 3 sodium ion. There is phosphorylation, which favors conformational change. There is the change in conformation. Now sodium is released, potassium binds. As soon as potassium binds, the phosphorus is released, dephosphorylation occurs, and it reaches its original conformation. This was the step 1, and this is very similar to step 1. Step 3 is equal to step 1. So in this case again, the potassium ion is released inside the cell and the cycle is repeated. That means it goes to this position. Correct? So this figure shows how the whole process is carried out in a single figure. So in this example that I have mentioned here I have shown the sodium exchange is used in epithelial cells in the gut to drive absorption of glucose from lumen and eventually into the bloodstream by passive transport. So this is just an example of how sodium potassium pump plays a very pivotal role in carrying out a very important process of absorption of glucose from lumen into the bloodstream. So you can go through the figure here as you see how the sodium potassium pump is aiding in the absorption of glucose, right? So this figure has been taken from the book Molecular Biology of Cell, the 4th edition. You can go to it if you want to have a broader scope of things. Again, about 1/3 of ATP in an animal cell is used to power the sodium potassium pump. That means it is an energy intensive process, right? Because 1/3 of the ATP that is present in the animal cell is required so that the sodium potassium pumps keep working. Okay, and in electrically active nerve cells, the sodium potassium gradients are used to propagate signals up to 2/3 of the ATP is used to power these pumps. So in nerve cells, about 2/3 of the ATP gets used up, and in other cells 1/3 of the ATP is used up in sodium potassium pump. So you can have a guess at how important the sodium potassium pump must be, that the energy currency of the cell, that is the ATP, is used so that the pump can be properly maintained. So with this, I conclude today's lecture. If you have any doubts, feel free to contact me. I shall be available to answer all your queries. Thank you for your attention.